Hello, everybody. Today, as part of our innovation showcase, we are going to talk to you today about the applications of organoid and organotypic cultures in infectious diseases, nephrotoxicity, and highly relevant cell-based assay development. We're going to have three speakers today: myself, Martin Stahl, Juan Hou, and Philipp Kramer. We are all scientists at STEMCELL Technologies based in Vancouver. And each of us are going to present a different organ system, including the intestine, the airway, and the kidney. I'm going to start today with speaking to you today about intestinal organoids and monolayer cultures. So before I get into that, I'm going to talk to you a little bit about how far organoids have come over the last 10 years. They've been revolutionizing in vitro cell culture models since their introduction over 10 years ago, and it all started in 2009 with Toshiro Sato and Hans Clevers when they introduced the simple mouse small intestinal organoid model. Since then, this has expanded to include most tissue types, including HPSC-derived tissues and primary tissues, covering most tissues in the body. Now, despite the advances that have been made under this subject, there's still a great deal that needs to be done, and researchers are working hard to advance even more directions. They're looking at how to create more complex models that include more cell types. They're trying to better recapitulate models in patient tissues and diseases. And what I'm going to talk to you today is how stem cell technologies can further the horizons of the field in these directions. So as far as intestinal organoids go, researchers are always advancing them as far as they can go. In the field of cancer research, in vitro cultures are recapitulating patient genotypes and phenotypes, leading to better treatments. For drug testing done by pharmaceutical companies, we are finding better and more representative models for drug toxicity, metabolism, and absorption in the intestine. And this is all bringing us closer to personalized medicine, something that manages to tailor our drugs and our medical treatments better towards our patients. Furthermore, intestinal organoids are being used for infectious diseases. This is using the enteric microbiota to mimic host microbe interactions, as well as looking at bacterial and viral pathogens. Now, of course, the most prominent virus that everybody's researching these days is the SARS-CoV-2 virus. And although the pulmonary system has gotten most of the attention as far as this infection goes, many SARS-CoV-2 infected patients also experience symptoms of gastroenteritis. This is due to the fact that the SARS-CoV-2 receptor ACE2 is highly expressed along the intestinal epithelium of the human intestine. Now, the question was always whether this ACE2 is also going to be mimicked by the intestinal organoids. And recent work by Zhu et al. And Lammers and Boehmer et al. In the last few months has actually found that human organoids do in fact express ACE2. And not only that, they are actually susceptible to viral infection and replication, and therefore can be used as an effective model in studying this new disease. Now, before I get into what stem cell can actually do to help advance this field, I want to talk to you a moment about what we're trying to model. And this is the intestinal epithelium. Now, the intestinal epithelium is made up of repeating units called crypts. At the base of each crypt is where the LGR5 positive stem cells reside. Now, at the base of each crypt, these stem cells are maintained through high Wnt signaling and low BMP signaling. As these cells divide, they create more cells which become undifferentiated transit amplifying cells, which can further proliferate. As they migrate up the crypt and approach the villus, they will lose their Wnt signaling and get higher BMP signaling, and those cells will start to differentiate. A majority of them will become enterocytes, which are important for both barrier function in the intestine as well as absorption. They can also go into one of any number of secretory cell lines, including goblet cells, which secrete mucus, which protects the intestine, or enteroendocrine cells, which will release hormones and other factors which help regulate the intestine. Now, in order to grow these intestinal organoids, we are trying to maintain and replicate the stem cell niche at the crypt base. So they have high Wnt signal and low BMP signal. And what we managed to create is a simple intestinal epithelium that manages to replicate these intestinal structures of the crypt. This is where stem cells intesticult organoid growth medium, or OGM, comes into play. When we introduced this, it was to establish and maintain and proliferate intestinal organoid cultures. Now, while this has been quite effective, this medium has both its strengths and its limitations. Now, intestinal OGM is exceptional for the robust maintenance of human intestinal organoid cultures and can even be successful with some of the most difficult to maintain donor lines. Now, as with the original published formulation, there is certain limitations to this in that while we are so focused on maintaining the stem cell population, we can end up with organoids that are primarily comprised with undifferentiated LGR5 positive stem cells and transit amplifying cell populations. Now, this is problematic if the researcher in question is trying to look a little bit closer at some of the more differentiated cell types. So why would we actually want to get these differentiated cell types? So a researcher who's looking at enterocytes might be looking at nutrient or drug absorption or metabolism, barrier integrity, or innate immune responses in the intestine. They could be looking at goblet cells in order to study host microbe interactions or infection and immunity. And finally, they'll need to have enteroendocrine cells if they want to start looking at the interactions between the intestinal epithelium and the nervous system as part of the gut-brain axis, or looking into the regulation of the intestine through the release of hormones. Or enteroendocrine cells can also be involved in diabetes as they can play a role in regulating blood sugar. Now, with such an importance on differentiated cell types, of course, the field has started to come up with solutions of their own. This usually involves withdrawing Wnt and enhancing BMP signaling. Additionally, some publications have tried adding small molecules that can drive certain target cell types, such as enteroendocrine cells. Now, while these have met with some successes, they also present certain challenges. A sharp withdrawal of Wnt signaling can remove support for the stem cell niche, and that can result in a loss of proliferation, and organoids can quickly lose viability and reduce the window where they are viable for conducting experiments. Also, by using small molecules, you can end up skewing your population of cells into different directions and not get one that properly replicates the diversity that's present within the human intestine. So what we need is a balanced approach, but this can be very difficult to achieve. So this is where STEMCELL comes in. While we've established the intestinal organoid growth medium for the maintenance and expansion of organoids, we are now taking the next step, where we're introducing the intestinal organoid differentiation medium, or ODM. This allows us to take the organoids that have been established with intestinal OGM and differentiate them to create more functional cell types. Now, a brief overview of how this system works. Organoids are grown up with intestinal OGM present within a matrigel dome, and we can do one of two things with them. We can either differentiate them in place by replacing the medium with intestinal ODM, which allows us to create differentiated organoids, or we can remove the organoids from the matrigel dome and plate them onto a matrigel-coated surface plate and establish them into a monolayer culture. This monolayer culture can then be further differentiated through the use of an air-liquid interface. So briefly looking at how this system works for the 3D organoids. You'll see on the top left how it looks with intestinal OGM and having standard proliferating organoid culture. On the right is organoids that have then been further differentiated with intestinal ODM. You'll notice that the cell lining of the organoids is starting to thicken as the cells become more columnar, and they take on a more budding crypt-like structure. This also starts to favor the appearance of secretory cells, such as goblet cells and enteroendocrine cells, which are largely absent from the expanding cultures favored by intestinal OGM. But importantly with this system, it still maintains a small proliferating stem cell population that can maintain the organoids for a prolonged period of time. We can look at immunofluorescent staining in order to see this in a little bit more detail. On the left side here, we've got a immunofluorescent marker for MUC2, which is a marker for goblet cells and mucus, and Ki67, which is a marker for proliferating cells. On the top left, you'll see an organoid that has been grown with intestinal OGM. You'll see it contains quite a number of Ki67 positive cells, but is largely absent for MUC2, showing there's no goblet cells present. As that organoid is differentiated with intestinal ODM, it starts to appear a lot more goblet cells, But importantly, if you look closely, you'll still see a small number of Ki67 positive cells showing that some proliferation is still taking place. Furthermore, if you look on the right, you can see staining for chromogranin A, which is a marker for enteroendocrine cells, and KRT20, which is a marker for mature enterocytes. Both of these markers are largely absent when growing with test called OGM, but they become very much more prominent when the organoid is differentiated with test called ODM. Now moving on to the monolayer culture. What we are trying to do here is take the 3D organoids that have been established with test called OGM and open them up into a monolayer that is more amenable to much different types of experimentation due to the accessibility of both the apical and basolateral sides of the epithelial layer. It is first established as a submerged layer with media on the top and the bottom, as you can see in the middle panel. And then it goes on to be differentiated using an air-liquid interface. Now with this process, the monolayer will thicken, more goblet cells will appear, and a mucus layer will even start to appear along the surface of the epithelium. We can see this more by staining for the presence of MUC2 and goblet cells. On the left, you'll see a monolayer that's still submerged, and while it contains some goblet cells, it's still predominantly enterocytes. As is differentiated with the air-liquid interface differentiation, goblet cells start to increase in number, and over time, a mucus layer will even accumulate along the surface of the epithelium, similar to what happens in the intestine. We can see this also if we start looking at gene expression of these ALI differentiated monolayer cultures. Using our marker for stem cells, LGR5, and comparing it against our 3D organoid cultures grown with test called OGM, we can see a high level of stem cells within the proliferating OGM culture, which substantially decrease with the differentiation medium in the monolayer culture, but importantly, expression is still there because there's still a small population. Now, we look at KRT20, MUC2, and chromogranin A, we see some increase with just the establishment of the monolayer culture, and then substantial increases with all three markers for enterocytes, goblet cells, and enteroendocrine cells upon ALI differentiation. One feature of being able to use these monolayer cultures is the ability to use transepithelial electrical resistance to measure responses of the epithelium. Now, TEER is an effective measurement of monolayer integrity, thickness, and tight junctions between the cells. Currently in the field, Keiko-2 cells are the most common model that are used for these types of studies. Now, if you look at the graph on the right, you'll see that while Keiko-2 cells do provide a substantial TEER reading in ohms times centimeter squared, monolayer cultures that have been grown within tests called ODM provide substantially higher TEER readings than Keiko-2 cell cultures. And while these amounts decrease a little bit upon ALI differentiation, this is due to the increasing complexity of the intestinal epithelium as more secretory cell types appear and overall TEER values are decreased. Now, you may have noticed that there's actually substantial variation within these intestinal organoid differentiation medium monolayers. And this is actually due to the fact that they start to group by both patient specificity and intestinal region. Each donor groups slightly on this graph. And furthermore, you can see much higher readings with colon-derived donor samples rather than ileal-derived donor samples, which is common and also found within the human intestine. Furthermore, we can use these monolayers as a further model for ion transport. Currently, intestinal organoids are a prominent model used for studying cystic fibrosis research using the forskolin swelling assay. As you can see in the two panels on the right, the addition of forskolin will activate the cystic fibrosis transmembrane conductance regulator, pumping chloride ions into the lumen of the intestine and increasing osmotic pressure, causing the organoids to swell. While this is effective, these monolayers will actually allow us to use an Using chamber to measure more precisely. We can see in the monolayer culture here, CFTR activation using IBMX forskolin activation. And furthermore, we can see the inhibition of CFTR using the CFTR inhibitor 172. Now, if we compare these results against the Keiko-2 model, we can see the intestinal organoid monolayer model is much more responsive than the Keiko-2 model, which shows that although Keiko-2s can provide good integrity in a monolayer, doesn't necessarily mean that they are conveying full functionality. Now, I'd like to go back to where I started here, looking at the SARS-CoV-2 virus in the intestine and what IntestiCult can do and whether it provides the organoid models that we need. And if you actually look at organoids that are growing with IntestiCult, we can see that they are indeed positive for ACE2, both with the IntestiCult OGM for the expanding culture, as well as monolayer cultures that have been established with IntestiCult ODM, meaning that these can provide effective models for studying this virus and how it infects the intestine. So finally, to conclude, IntestiCult organoid growth medium provides reliable and robust expansion, even in the toughest organoid lines. The IntestiCult organoid differentiation medium provides a support for unique cell types and a more representative epithelium. And the IntestiCult organoid differentiation medium with a monolayer culture can open up the organoids and create a flexible platform that can fit the experiments that you need. So the future of the intestinal organoids is going to be with enhanced differentiation and flexibility as we can establish monolayers or air-liquid interface cultures. Thank you.
Thank you, Martin, for the great talk, and then thank you, Martin, for the introduction. Hello, everyone. Welcome to the airway organoid and the organotypic culture session. As what Martin introduced, I'm Juan Hou, Senior Scientist at STEMCELL Technology. So to begin this session with an overview, the respiratory system is a complicated but a beautiful tree-like structure. Its major function is gas exchange and protection against environmental insults. The airway, often designated by structure and order of divisions. The nomenclature is diverse in the field. It can be proximal versus distal, and it can be central versus peripheral. But at this presentation, I will refer them as large versus small. As illustrated in these slides, large airway aligned by ciliated pseudostratified columnar epithelial cells, indicated in the top right picture. The ciliated epithelium has a uniform orientation of cilia that beat in unidirection toward the pharynx. This ciliary action, together with the mucus layer produced by goblet cells, provides a mechanism for the continuous transport of contaminating or excess material out of the lung. Large airway often surrounded by cartilage and smooth muscle, and its diameter is more than 2 millimeters. While the small airway is less than 2 millimeters in diameter, it's characterized by a loss of smooth muscle and cartilage, but retention of a cuboidal epithelium that are still ciliated. The large airway has more goblet cells, while the small airway has more club cells. The small airway ends with the alveoli, which is the basic breathing unit of the lungs, where the gas exchanges take place. So the in vitro human airway epithelial cell culture using primary cells is a great model to study respiratory disease. To do this, you can obtain the airway epithelium cells from a variety of different sources. You can through a nasal brush, through biopsies, or cells processed from cadaveric lung. And many of these cells are actually available commercially. And one useful side of those cell sources is that you can obtain them from different donors, either healthy or disease donors such as COPD, cystic fibrosis, or asthma. So those cells can be put into the culture flasks to be expanded. Those expanded cells phenotypically are airway basal cells, a type of adult airway stem cells that are capable of both self-renewal and differentiation into ciliated and secretory cells. The airway epithelial cell culture has 40 years of history. The first milestone publication that using serum-free medium culturing the bronchial epithelial cells published in 1982. Since then, the technology evolved over the years from maintenance of those cells for only 1 to 2 passages, 2 extended passages, and most recently, this 2D culture system was demonstrated to be amenable for gene editing. Pneumocode EX Plus medium, developed by STEMCELL technology, is a serum and BP-free medium that supports the airway basal cell culture for extended passages. The ICC staining data in the middle of this slide showed the expression of the basal cell markers CD49F and CD271 widely expressed in the basal cells cultured in pneumocode EX Plus. The plot at the right side is comparison of the pneumocode EX Plus with other two conventional media, which demonstrated that the cells growing faster and can reach more population doublings for extended passages in pneumocode EX Plus media. So when those 2D expanded airway basal cells are seeded into the differentiation system, and then they can generate a great airway model in dish that mimic the in vivo human airway. So there are two ways to differentiate the cells. One is as a 3D organoids, in which the cells are seeded into matrigel dome. After seeding, the cells will expand into basal balls within 4 to 7 days. Then you can switch to differentiation medium. Those basal balls will polarize, luminize, and form a fully differentiated mucociliated airway organoid, demonstrated in the top right schematics. Alternatively, you can seed the cells into transwall insert that contains a polyporous membrane, which allows the nutrients from the medium feed the cells from bottom side, while the apical side of the cells exposed to air. This is why it is called air-liquid interface culture, as indicated in the bottom right schematics. This air-liquid interface culture can be called 2D organoid because it is planar, not rounded. You can also consider it as an unfolded organoid. It is an open culture system compared to the 3D organoid, which is a closed culture system. In the rest of this presentation, I will go through each culture method and then demonstrate the tools that STEMCELL technology provide to support each culture method and their applications. So first, I will start from a 2D organoid, which is ALI culture. So air-liquid interface culture has been the gold standard culture models in the airway field for a long time. In 1988, Whitecap chamber, the first version of the transwall insert showed in the top left picture, was first described by Whitecap and the Wu group. The unique part of this air-liquid culture method is it is easy to access to apical surface of the epithelia. You can add viruses, inhalation drugs, air pollutant to the apical side. You can also measure the cilia beating, ion channel activity, airway surface liquid volume, and mucus secretion. It is also amenable for co-culture system. So pneumocode ALI medium is a serum and a BP-free medium that differentiates the airway basal cells into mucociliated pseudostratified large airway epithelium at an air-liquid interface. The physiological relevance of the culture in pneumocode ALI is demonstrated by the ICC staining image with massive cilia generation in the first circled image and the MUC5AC positive goblet cell in the middle image and the basal cell in the bottom of the epithelium in the last picture. So the bottom panel in this slide is a comparison between pneumocode ALI and ultra-G-based formulation used in the field. The data demonstrated that in pneumocode ALI, not only much better morphology achieved with thicker epithelium and robust cilia generation, but also better drug response to the epithelial ion channel are achieved, which is demonstrated by the using chamber electrophysiology assay at the right bottom. At the beginning of the presentation, I mentioned that the large airway and the small airway contains different cell types. Also, different disease affects different region of the airway. So It is important to generate region-specific airway cultures to model regional specific respiratory disease. To achieve this, we developed pneumococcal ALS medium, which was just launched recently. This medium can differentiate the airway basal cells into mucociliary cuboidal small airway epithelium at air-liquid interface. As you can see from the histology sections in the bottom channel of these slides, compared to the large airway culture in pneumococcal ALI, which is thicker pseudostratified epithelium, the epithelium is much thinner as cuboidal morphology in the small airway cultures in the pneumococcal ALIS medium. So the top panel ICC data showed that the small airway markers SCGB1A1 and SCGB3A2 are in particular present in the pneumococcal ALIS culture, but not or much less in pneumococcal ALI culture. So we also compared our medium with a conventional small airway medium. The small airway culture in pneumococcal ALIS generates more ciliated cells and the small airway markers expression than the conventional medium. I really would like to show this beautiful data generated from Shiminsky group in this very recent publication. So they demonstrated that COPD, a small airway disease, can be modeled in pneumococcal ALIS. Compared to healthy control in the left, the small airway culture generated using cells from COPD donor demonstrated correct COPD phenotype, including elevated MUC5AC staining and the impacted cilia generation. So, okay, we talked about air-liquid interface culture for both large and small airway modeling. Now let's switch to the 3D organoid culture. Fully differentiated 3D airway organoids can be achieved robustly and consistently using pneumococcal airway organoid kit, a new medium that will be launching soon. The top figure is live organoid culture. Bottom is a confocal image for airway organoid stained with cilia marker AC tubulin in green and the goblet cells marker MUC5AC in red. Often the time, the organoids will demonstrate synchronized cilia beating that move the liquid and the debris inside the lumen to one direction, which is visible when observed under a microscope. So this demonstrated the functionality of the airway organoids. Most importantly, this airway organoid culture also demonstrates the ion channel function by phosphorylation induced swelling assay. The top right shows the phase contract image demonstrating the robust organoid swelling after 6 hours treated with the drugs. Using this phosphorylation-induced swelling assay, it was further demonstrated that CFTR function can be rescued by VX809 by 20% in CF donors, indicated by the bottom right bar figure. This phosphorylation-induced swelling assay also suggested that the airway organoid culture has potential for high-throughput drug screening for cystic fibrosis. To summarize the comparison between 2D organoid versus 3D organoid, the two models has their own pros and cons. The ALI culture has over 30 years of long history, so it was very well established, while the 3D organoid culture has a short history. The ALI culture is insert dependent, and while the 3D organoid is insert free but needs a matrix. One advantage of the 3D organoid is that it allows clonal assay. ALI cultures also allow electrophysiology readouts using Using chamber, which is low throughput but able to measure individual ion channel activities, while 3D organoid cannot measure individual ion channel activity but allow faster readout for the phosphorylation-induced swelling assay, therefore has high throughput potential. The biggest advantage for ALI culture is that it is easy to the apical site, easy to access to the apical site of the epithelia, as I mentioned previously. So this really makes this culture method as the golden standard for respiratory virus study. So speaking of virus studies, the respiratory virus can be inoculated from the apical side of the culture, and the evaluation of the culture can be easily done on several areas of the culture. For example, the information can be collected from apical side, such as mucociliary clearance, cilia beating frequency, tear measurement. And then the information can collected from the tissue, the cell. For example, the intracellular viral counts, and then measure the viral spread and ICC histological study. The information can also collected from the basolateral side. For example, measure the cytokine release and the LDH release. It is very important to note that air-liquid interface culture with a pneumococcal medium were used by China CDC Virus Institution to identify SARS-CoV-2. This discovery was published in New England Journal of Medicine in early January. In this publication, scientists identified the viral genome sequence, characterized the several aspects of the virus, and disclosed this valuable information. These discoveries enabled the subsequent test for treatment and vaccine development. Given the viral spike protein of SARS-CoV-2 uses receptor ACE2 and requires TMPRSS2 for its activation, many researchers in the field right now use the existing RNA-seq data to analyze these two entrant proteins expression. So in this publication here by AO's group, found that ACE2 and the TMPRSS2 expression can be seen in bronchial epithelial cells cultured in ALI using pneumococcal medium. The percentage of ACE2 and TMPRSS2 positive cells has been compared across different cells present in the bronchial region of the lung. These two entry proteins seems to be expressed in the highest in the type of a transient secretory cells, indicated as the group of secretory 3 in the left side graph, as well as ciliated cells with lower level of the expression low. So indeed, consistent with the RNA-seq analysis, our in-house data we generated recently showing the presence of ACE2 in large airway epithelium cultured in pneumococcal ALI, indicated by the ACE2 staining in pink in the left image. And interestingly, we also found the ACE2 also present in small airway epithelium cultured in pneumococcal ALI-S medium and in the right panel of the picture in this slide. All these results suggest that ALI culture generated in pneumococcal medium is a great in vitro model to study SARS-CoV-2. So in summary, pneumococcal medium system provide versatile culture solution for physiological relevant in vitro culture, in vitro human airway modeling. With the pneumococcal EX plus medium supports extended expansion at 2D culture. Pneumococcal AOK supports robust differentiation as a 3D organoid, and the pneumococcal ALI and the ALI-S to support regional specific differentiation into large airway and small airway respectively at air-liquid interface. So thank you for listening, and with that, I would like to end my talk here, and I'm happy to take any question later after our last speaker, Dr. Philipp Kramer's talk.
Yeah, thank you very much, Juan, and thank you very much, Martin. My name is Philipp Kramer, and I would like now to introduce to you kidney organoids. But prior to diving deeper into kidney organoid models, I would like to give you a brief introduction into kidney physiology. Humans are born with about 200,000 to 1.8 million nephrons at the day of birth. And those nephrons are really the functional units of our kidney and the workhorses, because they ultimately are composed of a glomerulus where the filtration of the blood takes place. The filtrate is subsequently transported into the proximal tubules, from there into the loop of Hendler, and then further into the distal tubules. where the majority of the reabsorption of water and other components happens. The filtrate that is about 1% that is not being reabsorbed is then further transported in the collecting ducts, from the collecting ducts through the ureter into the bladder, and finally excreted out of the body. So various labs around the world have established kidney culture systems by, for instance, taking primary cells and primary biopsies using progenitor cells from developing rodent models and growing those in vitro. However, with the recent discovery of self-organizing multicellular multilineage containing kidney organoids, it has really revolutionized the field, the way we currently can study kidney diseases, as well as develop treatments in the laboratory. And with that, I would like to highlight a little bit the benefits and the limitations of PSC-derived kidney organoid models. As you are aware, there are certain challenges in the field, for instance, with 2D monolayer cultures that are less physiological relevant, and they don't resemble kidney architecture at all. Some of the solutions we see using PSC-derived kidney organoid models is that, for instance, they establish self-organizing organoids with a typical nephron-like segmentation. Another caveat in the field is that nephron formation is only found during kidney organogenesis. It ceases to exist. These progenitor or stem cells cease to exist upon birth, and they can also not be reinitiated after injury and during its repair. However, starting with stem cells that really mimic the developmental pathways really allow for the formation of physiological relevant cells. Another limitation is the access to primary tissues. And you can imagine that if you are studying specific inherited renal disorder, then this is even more challenging to access. PSC differentiation models are therefore more practical because these are self-renewing, expanding cells that can be differentiated in any cell types, including the kidney system. And with nowadays CRISPR-Cas9 technologies and sophisticated protocols out there, it is fairly simple to generate and introduce mutations in the genome of these iPS or ES cells to then study a renal disorder. Last but not least, it is also difficult to culture certain kind of primary tissues that also result in a lack of heterogeneity of those. On the other hand, PSC-derived kidney organoids, they generate a variety of renal cell types. However, with all PSC to kidney differentiation protocols, there are some limitations. So one of the limitations is that kidney organoids resemble a more fetal kidney. And this is true transcriptionally as well as functionally. The kidney organoids, they functionally don't develop a vascular access and a single access. As I already mentioned on my previous slide, the kidney organoids, they cannot be passaged because stem progenitor cells have self-organization and proliferation potential exclusively during differentiation. And the last point is also, it is really important to develop standardized and reproducible protocols and media, which already starts with the quality of your stem cells that ultimately go into your differentiation assay, and then controlling the variability using a multi-stage differentiation protocol. So for the rest of my talk, I would like really to focus on and take you through examples in the literature, as well as going through our newly developed STEMDiff kidney organoid kit that describes in how we can overcome and what we can do to overcome some of the challenges on my previous slide. And with that, I would like to go into applications where HPSC-derived kidney organoids are currently being used or where we would like to see them used in the future. So certainly basic research is a main field, and this includes general and renal-specific cell biology, mesenchymal epithelium transition, or studying kidney development and function. Here, I would like to highlight a publication that came out of a collaboration between Melissa Little in Australia and Ton Rabelink in the Netherlands. So they demonstrated that it was possible to mature the glomerular, tubular, and vasculature in the presence of a functional vasculature. And how did they do that? So they transplanted PSC-derived kidney organoids into mice and then demonstrated that the host-derived vascularization is able to connect with a pre-existing endothelium network of the organoids. That brings me back to overcoming some of the challenges from my previous slides in terms of maturity, because they were able to demonstrate the presence of host-derived blood flow through glomerular structures within the transplanted organoids. As you can see here on the bottom right of my slide, which shows an immunofluorescence image, the group introduced intravenously a fluorescently labeled dextran into the animals, and then on transplantation day 14, they were able to detect this fluorescently labeled dextran in the transplanted PSC-derived kidney organoids. Another field where we see already adaptation of kidney organoid cultures is to study inherited renal disorders. And here I would like to focus on a devastating kidney disease, which is called polycystic kidney disease, or short PKD. This one is an inherited genetic disorder that causes the formation of fluid-filled cysts in our kidney. The group of Benjamin Friedman and Joseph Bonventre was able to introduce genetic mutation in the genes of polycystin 1 or polycystin 2, which causes PKD. Then they took these mutant and isogenic control lines, differentiated them into kidney organoids, and found that at the end of their differentiation, the gene-edited PKD lines were able to form large cysts in vitro, as you can see here on the Breifeld images on the right-hand side. This really resembles the phenotype seen in vivo and demonstrates how PSC kidney organoid model can be used to study inherited genetic disorders. Another application is infectious disease modeling, such as COVID-19. And here I would like to highlight a very recently published paper by an international collaboration between Joseph Benniger, Ali Mirazimi, as well as Nuria Montserrat, as well as two of our scientists, Martin Stahl and Ryan Conda from STEMCELL Technologies, who also contributed to this work. This international group demonstrated first that PSC-derived kidney organoids express ACE2, which we already heard is the key receptor for SARS-CoV-2 infections. Then subsequently, they generated SARS-CoV-2 particles, infected kidney organoids, and were able to see an increase in RNA. This demonstrated a replication of those virus in the kidney organoids, and they were able to take the supernatants and perform a secondary infection with a different cell line. And most importantly, they also used the same kidney organoid model system to demonstrate that human recombinant soluble ACE2 can reduce viral growth at the earliest stage of infection. So this really highlights the utility of kidney organoids as a potential groundstone that helps to start looking into treatments for our current pandemic. Another application of kidney organoid cultures is the technology development, which includes device development or organ-on-chip technologies. Here, I want to show you a publication from Luigi Morizan, where he has taken pluripotent stem cells, differentiated them into metanephric mesenchyme. These nephron progenitors were then subsequently seeded into microfluidic culture systems. And then he demonstrated that with high controlled fluid shear stress, the kidney organoids resulted with an enhanced vascularization and maturation, which again ties me back into some of the limitations you heard earlier. As seen here in these immunofluorescence images on the left-hand side, you see a very rudimentary vasculature under static culture conditions. However, once the high shear stress is being induced by fluid flow, these kidney organoids, they demonstrate an enhanced vascularization. Last but not least, we would also like to see kidney organoid cultures being used in translational research, which then includes precision medicine approaches for genetic diseases such as PKD, as well as nephrotoxic drug or compound screening. To highlight a publication here from Stefan Zanicki in Cell Stem Cell 2018, he was able to differentiate his PSCs in 96 or 384 wells in a fully automated fashion using liquid handling robots. This high-throughput screening compatible platform was then used to efficiently model tissue-specific toxicity and disease phenotypes. In terms of disease phenotypes, he looked again at PKD and screened various PKD modifiers, and finally identified blebistatin as a myosin 2 inhibitor to result in an increased cyst formation in his PKD mutant lines. This sheds a new light on the role of myosin 2 in PKD. Now I would like to switch gears a little bit and focus on our STEMDiff kidney organoid kit that is recently developed and now launched. The STEMDiff kidney organoid kit is a serum-free medium that promotes efficient and reproducible differentiation of human pluripotent stem cells into kidney organoids, which are composed of podocytes as well as proximal and distal tubules with associated endothelium and mesenchyme. The key features of our STEMDiff kidney organoid kit is its relevance because it produces human kidney organoids that model the developing nephron and associated endothelium. On this slide, I want to show you the formation of these typical nephron-like segmentations, starting with podocytes that are here stained in red, looking for a podocyte-specific marker podocalyxin. From these podocytes, we can see that the structures, they lead into proximal tubules that are marked with LTL here in green, and then further connected with distal tubules marked with E-cathephrine. We have not confirmed the presence of loop of Hendler or collecting duct cells, and this is pretty much in line to other published differentiation protocols. Beside the three typical compartments of these nephron-like structures, which I mentioned earlier, the podocytes, proximal tubules, and distal tubules, we were also able to demonstrate the presence of CD31 expressing endothelium cells, as well as stromal cells that are expressing MICE123, as well as vimentin, shown here in the immunofluorescence images on the right-hand side. The next slide contains a heat map that really demonstrates that the kidney organoids we generate in our differentiation assay exhibit the expected changes in the gene expression, starting with undifferentiated cells that are highly expressing OCT4, leading towards the expression of mesodermal markers, intermediate mesoderm and metanephric mesenchyme markers, towards more specialized kidney cell types that are highly positive for podocytes, proximal tubules, distal tubules, mesenchyme, and endothelium markers. The next key feature of the kit is really the simplicity. It minimizes culture manipulation with a two-stage culture system and easy-to-follow protocol. This protocol is shown here, and briefly going through it, it's seeding single cells of ES or IPS cell lines into a culture format of your choice, changing media of your maintenance media MTSR1 for 2 additional days until day 0, where the differentiation is being initiated by simply switching MTSR1 to our STEMDiff kidney differentiation media. After a total length of 18 days of differentiation and multiple media changes, customers would expect to have tubular kidney organoids fully established. On this next slide, I present you the efficient generation of kidney organoids using multiple human ES lines, here shown on the left-hand side with H9 and H1, as well as 2 of our in-house IPS lines, 1C and MU1. These kidney organoid cultures are showing day 8 cultures, and on the right-hand side, you can see day 15 cultures of the formation of multitubular convoluted kidney organoids. Furthermore, another feature of the kit is that it's reliable. It produces low experimental variability through an optimized formulation and rigorous quality control testing. And this has been hopefully demonstrated here in this graph. by demonstrating the efficient generation of kidney organoids compared to a homemade media that was published by Friedman et al. In 2015. Here you can see manual kidney organoid counts per 96 well using our kit, or as I mentioned, the homemade media. And you hopefully appreciate the consistently higher number of organoids across the multiple cell lines, as well as a lower variability of organoid counts within each line. Last but not least, I would like to draw your attention to the high throughput potential of this kit, because we were able to demonstrate reproducibly the generation of kidney organoids in 96 and 384 well formats. That is shown here on my next slide. These are tiled immunofluorescence images of whole well 96 wells of kidney organoids from various cell lines on day 18 of differentiation. The top panel shows you the expression of our podocalyxin LTL ecatherine, and the bottom panel demonstrates the expression of CD31 expressing endothelium cells. We have taken those 96 wells with kidney organoids and took them into a nephrotoxic drug screening assay by using an antibiotic with known nephrotoxic side effect called gentamicin. And we demonstrated in the top left bright field images that with increasing concentrations of gentamicin, we saw the reduction of the viability of these kidney organoids, either shown in the bright fields on the left-hand side or in the viability assay on the top right. Using a sublethal doses of 2.5 milligrams per milliliter, we were then able to demonstrate the expression of a kidney injury molecule marker KIM1 by either immunofluorescence, seen on the bottom left, as well as by a KIM1 expression ELISA, here seen on the right-hand side. To sum things up, I would like to go through the fact that novel technologies enabling modeling of human kidney development, inherited and infectious renal diseases, then functional kidney organoid assays present a new tool with higher physiological relevance, and that also overcomes challenges seen with monocell culture systems. Our STEMCELL kidney organoid kit supports highly efficient and reproducible differentiation of PSCs into kidney organoids in a high throughput compatible micro-well plate, which can be then used for nephrotoxic compound screenings. And future improvements of kidney organoid models will certainly focus on increased maturity, structural complexity, and functionality. And that goes hand in hand with the development of robust and highly reproducible culture media and protocols. With that, I'm also now at the end of my session. After I have demonstrated to you the utility of kidney organoids, I would like to thank you. We've also advertising some of our tools and resources on our STEMCELL Technologies website. With that, again, thank you very much from all of our speakers, and we are happy to take any questions you may have. Thank you. Okay, so this brings us to the Q&A session. I hope everybody can hear me now. I would just encourage everybody to just type in the questions you may have in the question and answers tab on the left-hand side of your chat box. And I'm just starting by reading out the first questions we got already. So there's one question this is maybe a more general one, but it came up during the intestinal part of the presentation, so I may ask Martin to answer this. I read the question out. So it's, how do you think about the limitation to mimic in vitro and microenvironment for organoid models? Martin, do you want to take this?
Sure, I'll try to take that one. So it's definitely a limitation that exists. Notably that a lot of the factors that are trying to support the stem cells are produced locally by certain cell types, and they're meant to only act like a few cell difference. So when we add it to our medium, they're globally acting on the entire organoid. So obviously, like, we don't get the same kind of microenvironments that you might get in the gut, but at the same time, a lot of the
cell-cell signaling still exists. So you actually still get some pretty good microenvironments that are still appearing within these organoids. So it's a limitation, but I don't think it's that bad of a one. And I think it's one that is very surmountable as we go forward in developing this technology. Okay, I think we'll go on to our second question. Or Philip, you want to introduce the second question?
Yeah, I just read out the second question. So this was a question regarding the pulmonary part. So is this protocol available on Stem Cell Technologies website? If not, when will it be available? Juan, do you want to comment on that?
Sure. So we talk about basically 3 protocols in my talk, in my session. So one is the expansion, how to expanding those airway basal cells, for both large airway and the small airway. The second is how to differentiate those airway the epithelial cell, basal cells at the air-liquid interface. And then the third protocol is how to differentiate those airway basal cells into the organoid. So the first 2 protocols are available from our website. And then you can go to our website or visit Stem Cell Technology booth exhibition at this conference to request those information. And then the third protocol, which is differentiation of the airway basal cell into mature cell type as a 3D organoid, is not available at this moment. But it will be available 1 month later. So stay tuned for the airway organoid protocol and the data available on the website.
Sounds good. Thank you, Juan. I have another question regarding lung organoids. This is related to COVID-19. Does ACE2 is expressed in secretory cells in your lung organoids or in the lung organoids?
That's a great question. So we're still working on that. Based on our preliminary data, the data we showed in this presentation, it looks like it expressed in the secretory cell type. And however, we need to do due diligence and then to test more antibody and also test the co-staining with this ACE2 with other secretory cell markers. So we're still working on that, but looks like at this moment is the secretory cells.
Mm-hmm. Thank you. There's another ACE2 related question and COVID-19. So for the ACE2 expression, do you test the COVID-19 infection? The ACE2 expression is on an air-liquid interface. So this is a question I believe came up during the kidney part of the presentation. So in terms of the published work I presented from Vanessa Monteil et al. And Cell STEMCELL that was recently published, they actually did not an air-liquid interface model to my knowledge. Instead they used a V-shaped 96 well plate to force aggregate kidney organoids and then culture them and then also subsequently infect them with COVID-19. And the ACE2 expression, so this is something for our kidney organoid kit. If that's related to our kidney organoid kit, that's something we certainly look into it by RNA expression as well as protein data. However, we haven't done, we have just preliminary results indicating that there is some ACE2 being expressed, but we will look into more detail into this. Then reading out another question is there also skin organoids coming soon? So obviously Stem Cell Technologies is very interested in generating multiple organoid models, and skin organoids are certainly also on our radar. However, I'm not sure if there's a skin organoid culture media or differentiation media available soon, but we can certainly get back to you to provide you more details. So then I see that there's 1 more minute left. I will just read out one more question. Do you have cardiac organoid solution in Stem Cell Technologies? So that's something obviously goes hand in hand with the skin organoids. That's also of high interest for us. However, we will have to see when when this technology is being developed and coming out. So then to answer any remaining questions, I guess we will get back to you directly. And then I would like to encourage you to visit our booth, also get in touch with us directly at the ICE SCR. And thank you very much for attending this innovative showcase. Thank you and goodbye.